Thank you for your introduction. Good afternoon, everyone. I'm really happy to have this chance to introduce our work about these digital twins of individual patients and how we use it for the predictive, preventive, and personalized medicine. I'm not going to really details about the experimental stuff, but I work from the beginning how we have these conceptions, and then we actually have our real tools to introduction our products. In this talk, I will mainly address is 3 questions. What is digital twins? I think these conceptions already be introduced very nice from the Professor Garrios. And I will just go directly to our conceptions about how we use it for the personalized medicine. The second question is, what are they good for? Third one is, do they actually work in the clinic? Yeah, sorry. So this is the digital twin. So actually, we both those conceptions to our own conception, used for the personalized medicine. So from here, you can see in the figure A, there is a little dot in the patients. This is, we think, this is a local sense of disease. Then we will use high-performance computational model that is integrate thousands of disease-related variables to contribute unlimited copies of these patients. Then you have a lot of patients in here. Then each twins will be treated with different of potential drugs. Then we can use this, simulate how these patients react to these each drugs. Then we can identify which treatment actually works in the digital twins. We think this one, the green one, will be the best therapeutic treatment. Then will be selected and tested in real life, maybe real patients. So this is our homepage about the Swedish digital twins conception. Most of this conception today we're discussing, you can find here. And you also can find other informations where you are interested. Welcome to explore it. So you may ask, why do we need the digital twins? In many cases, we know its medications is not effective. Why? We will think about why. And also, it will cause many patients suffering. It's not only patients suffering, and also about the cost for the health care, and also productive loss, and also drug discovery. So you will think about why its medication is effective for the patients. One reason is diagnosis too late. When the patients went to the hospital, maybe symptoms will be already it's too late. And another is we don't have enough early diagnosed biomarkers because we don't know which disease. And also because the the disease already have progressing silently for many years, maybe decades. This can lead to complex disease, and also made it more harder to treat effectively. So how the digital twins contribute? First, we will construct the digital twins with the computational method. Then we'll test, just like I before said, we will test each drugs on patients to simulate how these drugs affect the patients and how the disease is processed. Then we can find maybe drug targets to prevent this disease. This is the first step. Next step, we'll focus on early disease mechanism. Constructing digital twins allow us have the chance to predict, prevent, also treat this disease in virtually before the symptoms appear. So this aim is just find diagnose biomarkers and also find the drug targets for early prevention and treatment in the individual patients. For the digital twins, we need some data, give us high resolution information. I will use this Lego model to explain why it's so important. We know it's bulk data, give us a lot of information. It's just like the Lego blocks. They blend in together, but we lost all the details. It just gave us the average cells of information. But this single-seq, like we sorting the blocks by colors and shapes, but they give us the information about the single cells. We know the expression of each cells. Special data, they will give our more information that goes more deeper. We not only know the cells, each cells, we also know where they are. What are they in the tissues? Just like Lego, knowing each Lego pieces fit in this structure. So for this single sequence data, this high resolution data allows us to find which cell type may be driving disease, and we can find cell type specific biomarkers. Then we can find precise drugs maybe can cure the disease. We need a model, a disease model, how we can treat this, use this conception to real life, or maybe test. So we think arthritis is the best one. Why? Because it's not only, it's a muscular skeleton. He has also have the immune disease features. And also have a lot of single sequence data can be used, and we can also treat it in the mouse model. So that's why we choose the arthritis as a model. We analyzed cell-specific genetic markers from 45 human cell types that enriched in the genetic variants. We found 24 cell types significant associated with arthritis. And also we found that there's 1,000 of genes related to the arthritis. We also used another, we built one, it's a multi-organ, multicellular model based on the single sequencing from mouse model. And we found over 1,000 inter-organ connections. You can see not only joint, lung, muscle, skin, and spleen, they were linked together. So it's in the symptoms, we only found it's symptoms in the joint, but it's not limited in the joint. Maybe others also have, but it's not have the severe, obvious symptoms in the other tissues. So based on all of these detailed information, we just understanding the disease. How can we go further to identify treatment? So do we have the actually method to construct digital twins. And how can we construct digital twins to prevent the disease process? Here's I will introduce a simple model. Here you can see there's a blue dot and a white dot. The white dot is a protein. When this white dot protein, we found it's connected with the disease, it turns blue. You can see in the figure, some of the blue dots scattered in this network, but some clustered together to form a module. And we found this module contains mainly important information, for example, pathways, biomarkers, and also can be the drug targets. But this is just a simplified module. In real life, most data is noisy, and mapping may be not so accurate. And we know it's finding early biomarkers is a challenge. But this module gives us a chance to link the disease and the genes in a systematic way. This is one articles. Our articles show that this module-based approach can be used to find biomarkers and drugs. So can we extend this idea to other molecules? For example, mRNA and the DNA. Yes, we can connect them together. But in real life, we make a diagnosis not only based on lab. Even best lab test is not enough for the diagnosis. So can we use others? For example, symptoms. I think symptoms are also important. For example, arthritis. The patients not only have the joint inflammation, they also have other symptoms, pain, and also fatigue. And we found that these symptoms tend to co-occur and form symptoms. That we can combine this symptoms module with the molecule module to predict maybe powerful predictive markers. Of course, we can extend the other layers to make these digital twins broadly across other diseases. We can include other layers. For example, lifestyle social factors and other factors. We can combine all these modules together to identify powerful predictive markers and drugs. For example, there's have a red dot. They are from the symptoms module. And then this blue dot from this molecule subtype. If we combine all this together, we can find one powerful biomarkers and drugs. We think this combination can be used for the personalized medicine. This is most from the one cells or cells information. So can we combine with the cell typing? Yes, we did. We combined this module with the cell typing, build the multicellular disease model. This model gives us not only interactions in the cells. You can see the interactions between cells. This will give us more information about how this cell type interactions with the disease. Then we can, based on this module, to map with the thousands of drugs in the drug bank. And we can rank these drugs based on the intracellular and intercellular centrality to rank these drugs. One practical example is our drug prior, our tools. We, based on the arthritis mouse model, built the multicellular model and mapped this module with the drugs. And we found the important high-ranked drugs in the activity cells, B cells. Yes, this dot is different cell types, and this color is similar, means it's the same cell types. And we want to validate this one, so we choose the top 5 drugs. Validate first in the human B cells. We found 2 of them, adapalene and amorine. You can find that they are inhibiting the human B cells. For example, activation, proliferation, and antibody production. And we did it also in the mouse model for the amorina. You can find that also the symptoms being induced. For example, the pore swelling, and also the joints inflammation. We did another validation. This is 2 patients. One is TNF responder, another is TNF non-responder. We used then sequencing data. First, build the multicellular model. You can see these dots as different cells. You can from here see they are totally different. For the TNF responder, you can see this green, orange means TNF. They have more central roles in the MCDM, multicellular model, but the TNF non-responder is not the central roles. And we map this multicellular model with the drugs again. We found the top-ranked drugs between 2 different disease patients are totally different. So this highlights how our MCDM-based approach can enable, predict, and prioritize personalized drugs, and shows we have strong potential to fund the potential drug treatment, personalized treatment. So can we use digital twins not only treat, but others to predict and prevent before the disease starts? We know this MCDM have limitations because they involve the subdominant genes. Even after prioritized cell typing, still include hundreds of genes. So we have another approach. We can identify the important genes that can regulate other genes. We use the knowledge by biological knowledge to identify these important genes. We call it upstream regulators. These regulators not only regulate one gene, they can regulate many genes, even include different stages. And validate it works. There's another approach. We used spatial data and combined with disease stages and also machine learning. We used a method, it's called studio time. We analyze this data and also arrange cells along with the timeline of cancer process. We found this sum of genes can link with the early disease changes. It's very interesting that we test those drugs, biomarkers in the 200 or I can't remember, 2,000 samples. We found it's a very high AUC. They can separate the patients. So all this approach shows the real promise for us. We can find the early diagnosis just using a routine clinic test. So summary, it's a network tools can be used for construct digital twins to predict and prevent disease. So thank you for all the founders and my colleagues and other collaborators. Thank you for listening.
Thank you for this interesting presentation, and this is open for discussion now. Are there questions, please? I have one. So you also discussed that you have often with proteins or genes that you have these network modules that light up. So it was with the blue and the
Yes.
white dots. So what are you doing if you have many networks or there are non-networks detected? So does this exist, and how are you then dealing with this situation?
You mean there's no connection between the dots?
That you don't find a module in certain conditions?
Actually, we didn't have that situation. That's a great question. Yes, it's maybe have some times if you don't have, that means maybe this module is not suitable for that. We may need to have or we need to have other cutoff, because this is data. It's really not really simple, just like this one. Because sometimes, because it's data noise or map is not accurate, then you can induce it. There's no connections. This is based on the PPI work, is protein-protein connections.
Okay.
So it's should proteins have the connection, but maybe it's difficult to find this perfect module. But how we can find this perfect module? There have a different cutoff. We can set up more selective or more strict.
Yeah. Okay.
Maybe I can also ask, I didn't quite understand how you get your input data and then either create or match to the network models that you have. Can you sort of reiterate what in practice you do to put together sort of the input data and then link it to the network?
This is we used is a differential, not differential genes. Is we used the proteins, the protein-protein network. Mm-hmm. There is a background, and how we can find it. These genes can becomes blue. We not only use one method to it. For example, these proteins they have MI, and this have a report be showing this MI have be linked to, for example, with arthritis. Then we use it become blue, and maybe also have a genetics. This also can link to the proteins. Then we can also blue. So this input is not only one side of molecules. There's a lot of different.
Okay. Yeah, if there are no further questions.
No. Yeah, thank you for both speakers for these excellent presentations. And I think it's certainly the future that we will have more in silico models and some digital twins that will guide us not only for our research, but certainly also in clinical practice. Thank you so much for this information.